friends, I am Romila Lemos, Assistant Professor, Department of Forensic Biology, Institute of Forensic Science, Mumbai. Today's topic of our discussion and learning is all about Forensic Serology Part 3. We will be discussing the following modules. Module 1, Introduction to Serological Techniques. Module 2, Enzyme-Linked Immunosorbent Assay, also known as ELISA. Module 3, Immuno Chromatographic Technique. Module 4, Importance of Immuno Chromatographic Technique. Module 5, Law of Individuality. Module 6, Precipitation Based Technique. Module 7, Conclusion. Traditionally, the detection and measurement of the antigen-antibody binding reactions serve as the basis of forensic serology. These assays fall into two categories, that is, the primary binding assays and the secondary binding assays. Recall that the primary binding assays involve the initial binding between a single epitope of an antigen and a single binding site of an antibody. Therefore, they are very sensitive. Although the secondary binding assays are less sensitive than the primary binding assays, they are easier to perform than the primary assays. The secondary assays consist of precipitation based and agglutination based assays. Precipitation based assays have been used for species identification. Agglutination based assays are more sensitive than precipitation based assays. These detect antigens located on the surface of cells or carriers and are normally applied to blood group typing. Recently, emerging techniques such as RNA, proteomics and DNA methylation assays and fluorescence as well as Raman spectroscopy can potentially be used for the identification of body fluids. ELISA is an antigen-antibody reaction based assay. In 1971, ELISA was introduced by Peter Perlman and Eva Engvall at Stockholm University in Sweden. It is a common laboratory technique which is usually used to measure the concentration of antibodies or antigens in blood. ELISA is a plate-based assay technique which is used for detecting and quantifying substances such as peptides, proteins, antibodies and hormones. An enzyme conjugated with an antibody reacts with a colorless substrate to generate a colored product. Such a substrate is called as a chromogenic substrate. A number of enzymes have been used for ELISA such as alkaline phosphatase, horseradish peroxidase and beta-galactosidase. Specific substrates such as orthophenyldiamine dihydrochloride for peroxidase, para-nitrophenyl phosphate for alkaline phosphatase are usually used, which are hydrolyzed by the above enzymes to give you a colored end product. ELISAs are typically performed in a 96-well polystyrene plate. The serum is incubated in a well and each well contains a different serum. A positive control serum and a negative control serum would be included among the 96 samples being tested. Antibodies or antigens present in the serum are captured by the corresponding antigen or antibody coated onto the solid surface. After some time, the plate is washed to remove the serum and the unbound antibodies or antigens with a series of wash buffer. To detect the bound antibodies or antigens, secondary antibodies that are attached to an enzyme such as peroxidase or alkaline phosphatase are added to each well. After an incubation period, the unbound secondary antibodies are washed off. When a suitable substrate is added, the enzyme reacts with it to produce a color. This color produced is measurable as a function or quantity of antigens or antibodies in the given sample. The intensity of color or the optical density is measured at 450 nanometers. The intensity of the color gives an indication of the amount of antigen or antibody. The types of ELISA. There are basically 3 types of ELISA on the basis of binding structure between the antibody and the antigen. Indirect ELISA, sandwich ELISA and competitive ELISA. Let us look at the first one, that is the indirect ELISA. Antibody can be detected or quantitatively determined by indirect ELISA. In this technique, antigen is coated on the microtiter well. Serum or some other sample containing primary antibody is added to the microtiter well and allowed to react with the coated antigen. Any free primary antibody is washed away and the bound antibody to the antigen is detected by adding an enzyme conjugated secondary antibody that binds to the primary antibody. Unbound secondary antibody is then washed away and a specific substrate for the enzyme is added. The enzyme hydrolyzes the substrate to form colored products. The amount of the colored end product is measured by spectrophotometric plate readers that can measure the absorbance of all the wells of a 96 well plate. The second type of ELISA is sandwich ELISA. Antigen can be detected by sandwich ELISA. In this technique, antibody is coated on the microtiter well. A sample containing antigen is added to the well and allowed to react with the antibody attached to the well, forming an antigen-antibody complex. After the well is washed, a second enzyme-linked antibody specific for a different epitope on the antigen is added and allowed to react with the bound antigen. Then after the unbound secondary antibody is removed by washing, finally the substrate is added to the plate which is hydrolyzed by the enzyme to form colored products. The third type of ELISA is competitive ELISA. This test is used to measure the concentration of an antigen in a sample. In this test, the antibody is first incubated in a solution with the sample containing antigen. The antigen-antibody mixture is then added to the microtiter well which is coated with the antigen. The more the antigen present in the sample, the less free antibody will be available to the antigen coated well. After the well is washed, enzyme conjugated secondary antibody specific for isotype of the primary antibody is added to determine the amount of primary antibody bound to the well. The higher the concentration of the antigen in the sample, the lower the absorbance. An enzyme-linked immunosorbent assay is an immunoenzyme assay that can be used to detect and measure the antibody or antigen in question. The most common ELISA that is used in forensic serology is the antibody sandwich ELISA. It is utilized to detect the prostate-specific antigen PSA to identify seminal stains and the amylase for the identification of saliva. An antibody, usually monoclonal coating, is formed by non-specific adsorption onto a solid phase such as the wells of a polystyrene plate. A sample containing the antigen to be tested is then added and binds to the solid phase antibody. Subsequently, a second antibody is added to form an antibody-antigen-antibody sandwich complex. The second antibody binds to different epitopes of the antigen. Next, an enzyme labeled antiglobulin is added to bind the sandwich. Subsequently, the excess enzyme labeled antiglobulin is removed by washing and the bound antiglobulin can be detected. The reporting enzymes catalyzes the substrate and produces colorimetric or fluorometric signals. The intensity of the signals can be easily detected spectrophotometrically and is proportional to the amount of bound antigen. The amount of antigen can be quantified by comparing the standard with known concentrations. Recall that antiglobulin can recognize an epitope that is common to all isotopes within an antibody class. The enzyme labeled antiglobulin is a universal reagent regardless of the antibody used as long as they are of the same class. Alternatively, antibodies in a sample can also be detected and may be quantified by an ELISA system in which the antigen is bound to a solid phase instead of the antibody. After an antibody in a sample binds to the solid phase antigen, an enzyme labeled antiglobulin is added to bind to the bound antibody. The bound antiglobulin can be detected and measured by the addition of an enzyme substrate. The enzymatic catalytic reaction is similar to the antibody sandwich ELISA procedure previously described. Immunochromatography is one of the most important and effective techniques in the detection of species identification in serology. These assays are also known as lateral flow test or simply strip test, which are the devices intended to detect the target analyte in sample without the need for specialized and costly equipment. This technique is the logical extension of the technology used in latex agglutination test, the first of which was developed in 1956 by Singer and Plotz. In this technique, a dye-labeled monoclonal antibody is contained in a sample well and a polyclonal antibody for the antigen or a second monoclonal antibody to a different epitope of the antigen is immobilized onto a test zone of a nitrocellulose membrane. An antiglobulin that recognizes the antibody is immobilized onto a control zone. The assay is carried out by loading a sample into the sample well. The antigen in the sample binds to the dye-labeled antibody already in the sample well to form an antigen-antibody complex. The complex then diffuses across the nitrocellulose membrane until it reaches the test zone. The antibody immobilized at the test zone traps the antigen-antibody complex to form an antibody-antigen-antibody sandwich. The presence of the antigen in the sample results in a colored vertical line at the test zone. The immunochromatographic device also utilizes a control zone to ensure that the device works properly and that the sample has diffused completely along the test strip. Unbound monoclonal antibodies diffuse across the membrane until they reach the control zone where they are trapped by the immobilized antiglobulin. This antibody-antiglobulin complex at the control zone also results in a colored vertical line. The test is considered valid only if the line in the control zone is observed. The presence of an antigen results in a line at both the test zone and the control zone, while the absence of an antigen results in a line in the control zone only. This method is rapid and simple and thus can be used as a screening test in laboratories and as a field test at crime scenes to identify different body fluids such as semen, saliva and species identification as well. However, a false negative result may be obtained if a sample contains a very high concentration of an antigen. Under this condition, the dye-labeled antibody will become saturated with the antigen to form antigen-antibody complexes. The unbound antigen will diffuse along with the antigen-antibody complex towards the test zone. At the test zone, the unbound antigen will compete with the antigen-antibody complexes for the antibody immobilized at the test zone. Since the antigen is in excess, the unbound antigen binds to the immobilized antibody, preventing the formation of the antibody-antigen-antibody sandwich. The resulting reading appears as a negative result. This artifact is known as the high-dose hook effect. To prevent the high-dose hook effect, a smaller portion of sample can be applied, or the sample can be diluted to reduce the amount of antigen applied. The benefits of an immunochromatographic test include: first, they are user-friendly in format. Second, they take a very short time to get the test result. Third, they possess long-term stability over a wide range of climates. And fourth, they are relatively inexpensive to conduct. The law of individuality states that every object, natural or man-made, has an individuality which is not duplicated in any other object. This principle at first sight appears to be contrary to the common beliefs and observations. That is, the grains of sand or common salt, seeds or plants or twins look exactly alike. Likewise, man-made objects such as coins of the same denomination made in the same mint, currency notes printed with the same printing blocks one after the other, excluding the serial number, and typewriters of the same make, model and batch appear to be indistinguishable. Yet, the individuality is always there. It is due to small flaws in the materials, in the arrangement of the crystals, imperfect sampling, or due to inclusions of some extraneous material. This individuality has been verified in certain fields. The most extensive work has been carried out in fingerprints. Millions of prints have been checked, but no two fingerprints, even from two fingers of the same person, have ever been found to be identical. DNA fingerprinting is another prime example of individuality of all living beings. The DNA of an individual is unique, and it is based on this uniqueness that the DNA evidence is now considered of a great evidentiary value in the court of law. The law of individuality is of fundamental importance in forensic science. Anything and everything involved in a crime has individuality. If the same is established, it connects the crime and the criminal. These techniques are based on the precipitation reaction and are used primarily for species identification in forensic laboratories. Let us start with immunodiffusion. Immunodiffusion is a passive method in which an antigen or an antibody or both are allowed to diffuse and therefore form a gradient from low to a high concentration. This is established for an antigen or an antibody or both. As a result, the precipitation occurs due to the interaction between an antigen and an antibody. The assay can be carried out in a liquid or a semi-solid medium such as agarose gel. The semi-solid medium can stabilize the diffusion process and reduce interference such as convection, which is the movement of molecules within the liquids. There are basically two types of immunodiffusion techniques. They are single diffusion and double diffusion. Let us talk about single immunodiffusion. A concentration gradient is established for either an antigen or an antibody in this particular technique. One serology technique based on this principle is radial immunodiffusion, a single diffusion method in which a concentration gradient is established for an antigen. The antibody is uniformly distributed in the gel matrix. The antigen is loaded into a sample well and allowed to diffuse from the well into the gel until a precipitation reaction occurs. The precipitate ring around the well is observed. The area within the ring of precipitate is proportional to the amount of antigen loaded in the well. Standards using known concentrations of antigen can be included in the same assay along with the samples, and a standard curve can be plotted. The amounts of antigen in samples can then be quantified by comparing the results with the standard curve. The second type of assay is the double diffusion, in which a concentration gradient is established for both and an antigen. Repeat, for both an antigen and an antibody. The most common examples are the ring assay and the Auchterlonie assay. Let us talk about the ring assay. In this double immunodiffusion assay, an anti-human antibody reagent is placed at the bottom of a test tube and a blood stain extract is placed on top of the bottom layer. The procedure involves sample preparation and extraction. A portion of the stain is extracted with saline at 4 degrees Celsius overnight. The controls include a positive control, known human serum sample, and a negative control, which is the extraction blank. The loading of the sample and antibody includes spinning of the anti-human antibody in a microfuge and transferring the supernatant into test tubes or capillary tubes depending on the volumes of the stain and the antiserum extracted. The sample is then placed carefully over the top of the antiserum solution, which is usually denser than the sample. The immunodiffusion reaction is done in the following ways. The reaction is carried out at room temperature. In a positive reaction, a white precipitate between the two layers can be observed after several minutes. This indicates that the sample is of the human origin. No precipitate is formed if a blood stain extract is from a non-human origin. In a positive reaction, a white precipitate between the two layers observed after several minutes indicates that a sample is of the human origin. If the blood stain extract is not human, no precipitation should occur. On occasion, more than one precipitin band will appear, indicating different reacting components with different diffusion coefficients. A standard operating procedure would be to include the necessary positive and negative controls with a question sample. The second kind of double diffusion assay is the Auchterlonie assay. The Auchterlonie assay is named after the Swedish immunologist Eugen Auchterlonie, who developed it. This assay can be performed in an agarose gel supported by a glass slide or polyester film. Wells are created by punching holes in the gel layer at desired locations. Often, a pattern with 6 wells surrounding a center well is used. The antibody is loaded in the central well, while the question samples and the controls are loaded in the surrounding wells. The double diffusion of the antigen as well as the antibody from the well is allowed to occur during incubation. If the reaction is positive, a precipitate line between wells can be observed at the end of incubation. The precipitate can be stained, enhancing its visibility to aid the observation. A single assay can compare more than one antigen to determine whether the antigens in question react the same way or differently with the antibody. This method is sometimes used to determine whether samples have come from the same or different origins. The procedure for Auchterlonie assay is as follows. The sample preparation and extraction include cutting out a small portion, that is approximately 5 into 5 millimeters of a stain or a swab. At room temperature, this stain is extracted in 100 microliters of water for about 30 minutes. The extract can then be diluted if necessary. Alternatively, a very small piece of the stain or swab can be inserted directly into the well. The controls include a positive control, which is known serum, and a negative control, which is the extraction blank. Substrate controls, that is the extraction of substrate from unstained area, can also be used if necessary. The agarose gel preparation is done as follows. A suspension of agarose 4% is heated until liquefied. The solution is then cooled in a water bath at 55 degrees Celsius, and the agarose is then poured on to a piece of glass slide and allowed to solidify to a thickness of about 2 to 3 millimeters. The wells are then punched, consisting of a central well surrounded by 4 wells using a template. In a positive reaction, a line of precipitate will form between each antigen well and the antibody well. This assay can also determine the similarity of the antigens. During the diffusion process, different antigen-antibody complexes migrate at different rates. Consequently, a separate line of precipitate will appear in the gel for each antigen-antibody complex. In an assay in which 2 antigens are loaded in adjacent wells and an antibody in the third well, the following results can be observed. First, if the 2 antigens are identical, the 2 lines will become fused. This phenomenon is referred to as identity. If the 2 antigens are totally unrelated, the lines will cross each other but not fuse. This is known as non-identity. If the 2 antigens are related, that is, they share a common epitope but are not identical, then the lines will merge with a spur formation. The spurs are continuations of the line formed by that antigen due to its unique epitope. This phenomenon is known as partial identity. Thus, in a species test, a positive result is noted when the precipitate lines for the positive controls and the samples fuse. No spur formation should be observed. Incubation at a cooler temperature is a slower process but has the advantage of producing sharper lines and sometimes additional precipitant lines due to reduced solubility at lower temperatures. Serological assays involving antigen-antibody interactions are one of the most important aspects of forensic serology. Apart from identification of the body fluid with the help of the antigens present in them, it also aids in the identification of the species from which the fluid originated. This kind of observation is important in excluding unwanted evidence samples as well as in cases related to non-human forensics. Although a number of labs employ direct DNA profiling for the sake of species identification, serological assays are still widely used in some of the forensic labs in the country. With all this information, here we come to the end of today's lecture. Do keep in mind what we have discussed today. It's time for you all to do some self-study. This is Romila Lemos signing off. If you want to learn more and enhance your knowledge, you may log on to our website for MCQs, quizzes, LORs www.cec.nic.in. Till then, goodbye.